Dr. Ianchulev is the inventor of the first ophthalmic intraoperative biometry device, which dramatically improved the outcomes of cataract surgery with more than 500,000 patients treated to date. More recently, he founded two companies in the field of ophthalmology, Eyenovia Bio, that develops high-precision microtherapeutics, and Iantech, a micro-interventional medical device company. Dr. Ianchulev is a prolific researcher with 82 publications and over 3,500 citations. His current functions include clinical associate professor at UCSF, CEO, Chief Medical Officer, Managing Director at a private equity firm, and a venture partner and board member. Please welcome Dr. Ianchulev, who will give the keynote presentation on innovation through the eyes of an eye doctor.
Thank you, and you did a brilliant job pronouncing my name. So it's great to be here, and thank you for inviting me. I'm a recent transplant to the New York area. I spent most of my time in California, the last 20 years, the last 15 in the Bay Area. So I came here with a little bit of apprehension because I thought I would miss the innovative spirit and what we have, which is quite special in the Bay Area. But that's not the case. Actually, over the past 9 months, I've been working very closely with the group here at Mount Sinai, which I thought was one of the most progressive groups, and I've been very lucky to actually join Eric and Sybil and everybody else to be an entrepreneur in residence, which is my excuse to have a couple of hours of enjoyment and really seeing and witnessing how the technology transfer office works here. So I figured probably most of you hear a lot about cancer. You hear a lot about genetics and biomarkers and immunology. And I know I did too when I was at Genentech, which was a cancer and biology and immunology company. But at the same time there, we developed something very special, which was one of the really successful drugs and biologics in ophthalmology. And as my oncology colleagues discovered, there was a lot about ophthalmology. And I would like to take a little bit of time, and thank you for all coming here to hear the story of I call it eye doctor with an eye, because as an ophthalmologist, I've spent a lot of time treating patients, but probably over the last 10 years, I spent a lot more actually on the eye side, which is really innovating and trying to develop treatments that work with patients. Again, these are my disclosures. Again, I've been really blessed because I know the failure rate of everything we do in innovation is pretty high. And I remember when I was at Genentech, when we looked at the portfolio, even for a biotech company, we kind of assumed 90% of the things we innovate are not going to be in the clinic. And it's very challenging because how do we validate targets when there are no great models? How do we rationalize biology when we don't truly understand it? So I think these are great terms and we're striving. I've been lucky because actually most of the things I've worked over the past 10 years did turn into treatments for patients. And then I was actually next door listening about how we discover drugs in psychiatry and depression, and it hit me that, you know, when I look at my patients and the ones where we deal with blindness, what is common among all of us and blind people is that we spend most of our lives in the dark. We're trying constantly to discover, how do we get a new treatment? How do we find ourselves? What should we do with our lives? And even though we see, we're actually most of the time groping around. And just like some of my blind patients, we're constantly looking in the dark until we direct the torchlight and we actually start seeing shapes that turn into objects. And they turn into something that has meaning to us, and eventually things that we translate and find purpose. And again, blindness is a horrible thing for most patients. But if you talk to blind people, many of them say that they're not deprived of any senses. They've developed their senses and they see things, they see people, they see a lot of things. Even some of them have escaped, and there are great stories about that, how they've escaped what happened on September 11th, and were able to find their way out of the towers better than some people were saying. And so for me, what I wanted to briefly touch upon is what motivates me to develop and to work in innovating in ophthalmology. In fact, when we look at blindness, it's interesting. We talk about cancer all the time, and we talk about heart disease, but when you ask people what they're most afraid of, it turns out that blindness and paralysis are really on top of the list. And they certainly would be for me. I simply didn't know how it would be for other people. Obviously, they're all pretty disabling conditions, and we don't want to diminish the significance of cancer and heart attack. But at the same time, we do want to highlight that blindness and paralysis is really on top of most people when we do the surveys. And right now, in the United States, we have 7 million people who are blind, visually impaired, and about 700,000 kids to 20 years of age. So it's quite prevalent, and we see it all the time. And really, for me, having been involved in a number of technologies, one of them, and I'll talk briefly about some of the stories, because every innovation is a story. We're building a story as we go along, and it doesn't happen over a year. I mean, when I first started out with some of the ideas that I thought were great and can be transformative, it just took a decade to really see the results. And that's what I would say, and I'll share some wisdoms from my own experience, but it is a story. You're taking on something where you innovate that's going to take you somewhere, and it's going to be a destination, and it's going to be a long path that will take us to that place. So I've been involved with several technologies that I want to share with you today, and they illustrate different aspects of innovation, which I thought may be helpful for anybody that really considers innovation in their career. One of them is a innovation in biotech, which is the Lucentis story, which was one of the major treatments turned into a major treatment for macular degeneration. Obviously, one of the main agents that causes the main diseases for blindness in this country. So it's a really, really impactful place, and we'll talk about this. The other one is the cataract, #1 surgical procedure in the world. There are probably about 25 million cataracts that are done in the world every year, and about 25 that are not done because people don't have access. And we innovated, and we came up with a infrared laser guidance technology, which greatly improved the precision of cataract surgery. And I'll share with you that story, because for me, in fact, it started in residency. So it really illustrates how even as a resident or as a medical student, you can start nibbling at something, and really, over time, you can build the story and see it through to patient care. And then the last one is another surgical innovation, which is a mixed device. It's actually a stent for ophthalmic use, and it's only the second stent that is approved in a very new, very dynamic field, one of the hottest fields in ophthalmology now, called MIGS, micro-interventional micro-incisional glaucoma surgery. It impacts a lot of patients, and I'll tell you about that. We just got that FDA approved a couple of months ago. Again, of the most projects I've worked with, I was lucky because most of them got approved. We saw Lucentis through the BLA, which was a really phenomenal path that really involved me and hundreds of other people. And that was great to see that right now it impacts a lot of patients. We also got the other two technologies FDA cleared and on their path to patients. And so when I spend time with my venture guys and with the bankers on some of those companies, what makes most impact for them is the numbers. You know, well, that Lucentis is making $4 billion a year. Well, that's a huge treatment. It's a blockbuster. That we sold one of my companies as where I was the inventor of the technology for $400 million, and we just sold another one for $700 million. So most venture guys love to talk to Sean, and they come to me and they, say, hey, what are you working on now? But in fact, for me, when I start out with a project, I really never think about that. I really never think as to how much money or what is the market. I know we constantly try to define the market, but just going back one slide, if you look at Lucentis, for example, in the hands of one of the biotech companies that did a lot of market research 10 years prior to its introduction, they never thought that this would be more than a $50 million per year drug. It actually, when you get through all the marketing research back in the days, they didn't believe it will make more than 50 million. And partially because people didn't believe you can treat a disease with intraocular injections every month. And so, despite all the great research, nobody could have foreseen that this is a $4 billion franchise now. So the way I look at it is, how does it impact me? How does it impact my patients? And ultimately, what makes me most happy right now is not the amount of money the drugs actually generate or the devices. It's actually going to all the meetings and say, Sean, I just used your technology and I nailed it. All my patient is very happy because I could solve the cataract. Or, we look at the numbers and we're seeing 300,000 patients that are treated every year with Lucentis, or 500,000 patients to date with the intraoperative aberrometry. So again, maybe because I'm a biased clinician, to me that really makes my day and really motivates me to do new projects. So when I looked at that, pretty much every one of these projects have something in common. My best preceptors and my best friends usually said, why are you doing that? It doesn't make sense. And I was discouraged along the way from day one, not because people wanted me to fail, it's because they wanted me to succeed. So I kind of put down for everybody in the audience a couple of my takeaways from what I've seen through the past 10 years. And number one, in terms of Dr. I's method, that's what some of my patients call me, to the madness, is I think for medical innovation, again, it's very nice to have a fully validated marketing model and understand what the market opportunity is, but I really haven't found that to work that well, especially in the early stages of drug and innovation and drug and device development. So for me, it's been really trust clinical instincts and intuition of clinicians, best if you're on. So I just happen to be a doctor and a clinician, and especially in the ophthalmic space, really treating those patients, so I can really have my finger on the pulse of that. But it doesn't have to be yours. I think you can listen to people that spend real time with the patients and with the diseases. Number two is really follow through and pressure test until the lid blows off, because it's very easy to make early on some assumptions or borrow some comments by people that are looking at a problem just from the spectacles that they have. So really pressure testing and really trying to understand where things are and where you want to go and where this technology will ultimately be is really important. Collaboration is important too, and I think everybody understands that, because today it's really hard to do things in the garage. It's not a one-man or a two-man enterprise, so it's not a garage initiative. It doesn't even take a village. Some people say, oh, it takes a village. No, it takes a tribe, because at the beginning, you cannot involve too many people, but you also cannot be completely solo. So you have to really find people and minds of positive conspiracy that will engage with you to help you crack that problem. And make sure you sign your confidentiality agreements, as Sybil would say. Surround yourself with best-in-class people. I've constantly exploited best-in-class people that are better than me. And I've told them, I need you to help me. Because frankly, when you're innovating, you're trying to develop something that hasn't been done. To me, it was quite obvious that you need to find people that are better than you or can help you solve difficult problems. Again, embrace critique and discard criticism. And that is very important, because a lot of people critique and a lot of people criticize, and sometimes the lines are blurred. Even when my wife sometimes gives me feedback, says, is that a criticism or is it a critique? So again, being able to take the critique because I feel critique is something that aids in the discourse, it pressure tests, versus criticism where many people would say something just because they discard it or because they have a different vantage point. Take calculated risks and try to get multiple bites at the apple. Again, doing everything that you want to do for some of those projects for me has been interesting. You try to do what's right, and very often people will say, well, we can only do that. And if you challenge people, you can sometimes do a little more, and you say, we'll do one more experiment, or we'll try to look at another variable or at another outcome. And it just so happens that that turns out to be the one that matters 2 years later. And there is all the good reasons of why you shouldn't do it, because you want to save resources. But again, being able to extend and stretch the rubber Band-Aid and get a little more, especially in the early stages, I think pays a lot of dividends at the end, because we don't know anything. So wherever you poke your finger in the dark, you may find something. And really trying to have multiple bites at the apple as you go out and look in the dark, I think helps. And then the last one is don't latch on to friendly advice about what's best for you and your future, because sometimes that's not what you should do. And again, I'll give some examples. Really, for me, that wasn't what many good friends gave me on all these 3 projects, as we'll discuss. They gave me very different advice than what I did, and ultimately, I succeeded because of what I did and not what I followed. So it always brings the quote here by Lincoln, which says, the best way to predict the future is to create it, because sometimes we just can't predict it. So again, outside this didactic slide, I want to go back to, again, some of my personal experience on the first project on the retina world. When I went into trying to develop Lucentis, Lucentis and the retina, the world of retina in ophthalmology was pretty barren. There had been nothing, no single therapeutic that was there, and it was very unique field where there were a number of retina specialists who were doing a lot of surgery, but they never thought about a therapeutic approach to their disease. So if you look at it from the outside, you'll say there is nothing there. And in fact, that's what my preceptors have shown. Are you really going to go into retina? Because if you want to do some innovation, you should go to glaucoma. Well, it so happened that really that was where the field that's witnessed the biggest therapeutic in ophthalmology. Then when it turns out, when I started diving into the cataract surgical technology, we'll talk about later, some of the colleagues that I worked with, in fact, in my residency program, they said, well, Sean, that will never work. Why waste your time? We already have the right tools that we can solve the problem as it is. And I just felt like we didn't have the right tools. And again, they all are now lecturing about the same technology 10 years later, and they say, you were right. So again, if I had taken their advice, I probably would be in a different place. And then when it comes to CyPass and the glaucoma technology, I'll show you later, when I did my initial research of whether I want to undertake this, most of my key opinion leader friends said, Sean, who needs that technology? Stenting in glaucoma is really not something that we need. And then I always remember my advice as a pre-med student when I was without a green card and I didn't have the opportunities here that most people did at the time, being a foreigner and immigrant. My pre-med advisor said, Sean, you should really think about it, because as a foreigner, it's impossible to get into medical school. It's really tough, especially if you don't have for most people who know some of the immigration challenges, there is no way you can make into medical school. Have you considered veterinary medicine or dentistry? So that being the end of that, I kind of realized early on that people may try to help you, but when it comes to innovating and finding the way in the darkness, whether it's a way for your own professional development or whether it's a way of how to solve a project or come up with something new, it's ultimately you are the expert and you will have to really pressure test it. So let's talk about Lucentis, which is the first story that I wanted to tell you about. It was by far one of the biggest innovations we've had in the field of ophthalmology, in our tiny little field that deals with the eye, which is one of the smallest organs in the human body, but as I like to say, it occupies a big part of your brain. And again, in retina, we had the fundamental problem that people who were affected by macular degeneration went downhill really fast. They lost vision, and they went from 0 to blindness in literally 2-3 months. It was a really a very, very disabling diagnosis to tell somebody they have macular degeneration. And what happens is, as you can see here, and I don't know if we can, okay, right here, is right in the macula, which is a single dot of about less than a millimeter, and it accounts for 90% of your vision. So if I were to shoot a laser in your macula, right in the fovea, a single laser burn can really blind you. Just in that, even though the retina is so big, but what is really dealing with your central vision is less than a millimeter, even less so. So again, it so happens that in macular disease, in macular degeneration, you have almost an explosion of inflammatory and new vascular processes that happen right in that unfortunate spot. And when people present, usually they say, well, you know, I look at the door, I looked at something one day, and I started experiencing these wiggly lines, and I feel the lines are not straight. And then within 3 months or 4 months, when we diagnose them with macular degeneration, they go from 20/20 to 2400 really fast. So it was a really disabling disease. And again, in ophthalmology, we are kind of the recipients. We're the orphan child. We do receive a lot of technologies that trickle down from other fields, usually from cardiovascular and from cancer. And when I was at Harvard, I worked in Judah Folkman's lab with Tony Adamis on anti-VEGF, which was really, for me, one of the first exposures to anti-VEGF treatment. And it so happens that anti-VEGF, which is Avastin, and right now we know some other therapeutics in cancer, they made a huge difference in cancer by blocking the vascular endothelial growth factor. And this is the factor that is responsible for the growth of new vessels. It's the same mechanism that happens not in a cancerous or oncologic process, but in this process of macular degeneration, where those new vessels grow, and the whole growth of this new vascular complex is driven by VEGF, by 80 or 90%. So if you use the same vascular endothelial growth factor antibody, you can block it, and you can really impact the disease course. And so Genentech worked for many years, and I would say, I wish I can say this is my project. No, I came in when we were developing it in a clinical way with patients, but this started in the labs probably 20 years prior, when the same antibody, which was Avastin for oncology, was miniaturized for ocular applications and became Lucentis. But the bottom line is that the binding site for this binds VEGF and neutralizes VEGF, so it doesn't cause the growth of new vessels where we don't want to see them, and it doesn't cause a problem with your vision. And so we reported, after I joined Genentech, before we launched, we came up with the data. It was one of these very amazing things where I still remember we were in the data room, and we don't know what we're going to find out. We have no idea. It's really a masked study. We don't want to call it blinded study, because in ophthalmology we don't like to call anything blinded. But we basically peeled the envelope, and when we saw it, it was just such a shock. I think it was like at 12 at night. And in fact, it was in a very remote place where it was under strict security, because obviously this was even material information for the company. And when we saw that, I said, wow, we don't need... I remember some of the sales and marketing guys, I said, why do we need sales and marketing guys? I mean, this would sell itself. There is no patient that wouldn't want to be on that drug, because here is what happens. These are two studies that were done in two slightly different variants of the disease. And the black line is what happens with the natural course of the disease if you didn't treat with Lucentis. And you would see exactly what I told you. Within a few months, their visual acuity, which is the letters of the vision chart that they count, which is our basically outcome instead of mortality or morbidity, you can see how quickly patients progress and they lose their sight. And in fact, many of them already start out with lost sight. Even here at baseline, they already presented with a disease, so they already lost probably half of that. And the rest goes really, really quickly. So if we didn't give you an intravitreal, meaning in the eye injection every month, you would be here, you'll be blind. You can't drive, you can't do your daily activities, you'll be in the dark. And it happens very quickly in the second eye. So it's an asymmetric disease, but it impacts both eyes, and then many patients just go blind in both eyes. But if we get you the Lucentis treatment, not only did we stop the progression of the disease, which would be we maintained you lost some vision, but we maintain you don't go blind. But in fact, we restored your sight. It's never been seen something like that in ophthalmology with such a dramatic disease. If that was cancer, it would be amazing. And again, this is the other study that showed exactly the same thing. You can see here the control group really crashed. They went downhill and they lost their vision in that eye very quickly. Whereas with Lucentis, not only did we prevent that, but we improved it. So it was a phenomenal therapy when it first came out, and it really opened up the space of where now there are millions of injections. And again, the unfortunate part is that you have to get an injection in your eye once a month or once every 2-3 months. The fortunate part is that most people don't have a problem with that. They don't feel it, and we've become really good at injecting your eye. Now, the interesting part about that project is, before you go out to develop and sink about $1 billion into this, let's say you validated your target, you've realized this is a great way to treat the disease, you go out and you ask a bunch of doctors and say, would you treat ever, assuming that you have a situation like that, would you ever treat a patient with monthly injections in the eye? And by that time, we had never done that. The only time you would inject somebody's eye with a drug is when they have a huge infection and you inject an antibiotic. So all of the market research came, are you kidding me? There is no drug. Even if it transforms what you do, there is no way we can be injecting somebody every month. So now we've reported data, I think, up to 5 years and 10 years, and guess what? Patients show up religiously, and it's actually not that bad. But if we go out and we survey, and one really stops at the results of that survey, I think it could have been a different story. One shouldn't have developed that drug. In fact, it was irrational to develop that drug. And it so happened that it didn't just transform macular degeneration. When I was there, we launched 2 other major diseases. One of them is retinal vein occlusion. About 1.5 million in America have retinal vein occlusions. Macular degeneration, 1.7 million. And then diabetic macular edema is 800,000. So it so turns out that a lot of retinal diseases, which are huge, like diabetic macular edema, Lucentis is approved for that now, are driven by the same vascular endothelial growth factor. And again, they're all being treated with intraocular injections. So that was really something that you don't see coming, and I think that's one of the things about innovation is as you start really working, you have to find one thing that excites you and one thing you're going to solve, in this case, blindness. And again, we figured out there were a lot of challenges. Would doctors use it? Would they inject patients? Would patients show up? Would reimbursement people pay for it? And guess what? They all come and together, I think eventually it's solved, as long as you solve the problem you're solving, which is, in this case, blindness. So that was one of the innovations which I thought was really, for me, was a privilege to be part of, and it was a phenomenal thing to experience, of developing a biologic in what, at the time at Genentech, they used to call one of the more complex and definitely the most expensive drug development programs, because having a bunch of ophthalmologists and surgeons treating this and being part of the study was very challenging initially, and it took a lot of determination. And that was part of what I inherited from my engagement, is that really in the company at that time, there was just a huge emphasis on science and unmet need. And that's the only way you can solve that, because, as I said, most people believe there was no commercial market and definitely no treatment ability if you do this. So I'll just switch gears now and go to the device side. I've actually been lucky because I've spent a lot of time both on the drug and device side, and it turns out they're very different. And the people that are involved on the device side are engineers, and they're very technical, they're very structured, and they want concrete answers all the time. Whereas the people on the biological research and the pharmaceutical research, it's controlled chaos, and there is a lot of brainstorming and a lot of kind of a research and discovery happening at the same time. But I've appreciated actually working with both. Has been really, I've improved my own skills and communication to be able to articulate the clinical need on both sides. But I want to show that for particularly the people in training, because this really was a project that was my resident project when I was in second year of residency, that we came up with the idea of really using intraoperative aberrometry, which is an infrared laser guidance system, to improve cataract surgery. And you can see here the early efforts of what we started out, because obviously we didn't have a budget. And what we ended up doing is we didn't have the device because it's going to cost a lot of money to build. We actually used the surrogate device from the clinic, which was used for a completely different application, and we were able to bring it into the operating room. So those pictures are how we started. So you always find out a way to start cheap because, you can't get the grants or you're too busy. But we started out with this, and then you'll see the device that turned out to be in the clinic. But it wasn't easy because I had to do my cataract surgery, and then before we implant the lens, we had to break sterility, bring the device, do the measurement, and then do the sterility again. So it was actually challenging from that perspective, but it generated a lot of data. So what was the problem we're trying to solve there? Well, cataract surgery is a really common procedure, and we all take it for granted for all good reasons because it's very successful. Once you have that, your vision is restored. But about 10 years ago, one of the things that we didn't really account for very carefully is that after you do your cataract surgery and we put an intraocular lens implant, about more than 30%, or in some cases more than 40% of people, that implant was not enough. It was so wrong that they still required glasses. So not only reading glasses, but glasses. So let's say if we take a person with glasses today and we do cataract surgery, we can get it so precise now that in 99% of cases, you won't need glasses for distance. But that wasn't the case about 10 years ago. And in fact, it was a pretty big number. Medicare didn't care in the pairs because they said, well, get them some glasses, and that's not a visual impairment. They can just walk around with glasses. And then even most surgeons at the time didn't care because they said, well, patients seeing well, they can get glasses. But we say, well, you still need to give them another lens on top of the lens you just implanted. It doesn't make sense. And today, with the intraoperative guidance and refractive system that we developed, that number is less than 5%. And it's probably even smaller with the very premium or very, I think, very technologically savvy practices. So that means that in 95 to 98% of the cases, you can have your cataract surgery and you don't need glasses anymore. And again, people and patients have learned to appreciate that because they have LASIK and they have other procedures, and they expect, especially in today's world where people over 60 are very active, they expect to see well and not to wear glasses. So we delivered on that. It started out with a resident project, and it took 10 years. And I remember every year I was talking, it was so funny, they still would let me talk at the Academy of Ophthalmology. And the first 3 of the 10 years, most people said, oh, that's interesting, but they didn't quite understand what we were doing. I was there last year, and I was teaching the course of that, and there were probably about 50 presentations on that technology in pretty much every zip code in the US. And I said, well, I don't have to say anything more because it's literally everybody's talking about it. But basically, it boils down to a paradigm shift. And how do we calculate the lens that you put in somebody's eye? Obviously, we don't all wear the same shoes or the same suits. You have to customize it. But now what we do is when you go to cataract surgery, we have an array of shoes and lenses, and we've looked at your anatomy of the eye, and we've predicted what lens we should put. Let's say a 20 diopter lens or a 21 diopter lens. Well, it so happens that when you do the prediction in the clinic before the surgery and you're using old technology, you can be only that good. But when we do the laser-guided infrared aberrometry, we can be way better. It's a completely different paradigm. It uses completely different technology. And you can see the current technology, the conventional technology, when you do it prior to the surgery in the clinic by measuring the length of the eye and then creating an estimation model, was about 50 years old. It came out from the Russians. And they did a great job at the time. And there were a lot of formulas. And every time I've learned when there's a lot of something rather than a single one, it's a good thing because that means there's not one thing that really works. And there's a lot. And all these formulas, but none of them were really good. And then we came up with the intraoperative aberrometry, which really changed the way. And again, it's interesting because if you look at the conventional technology, if you draw a curve of how conventional methods really improved cataract surgery, it's a very slow curve that kind of peaks here. But after the 1990s, it plateaued. So now you can get just another formula and another modification of that technology. But guess what? It's not going to give you this. It's going to get you from here to here. it still doesn't solve the problem, just because of the limitation of conventional biometry. And so there were a lot of advancements, but ultimately what we realized, we needed a new paradigm, a completely different approach, which uses the infrared aberrometry, never used in ophthalmology. It uses it inside the operating room, which means the first biometry in standard cataract surgery. And now, within 1 year, we pretty much were at the time, just as we were developing, we were pretty much better at that. So we use a new paradigm, and we're able to now achieve better outcomes, and now we've exceeded them with that technology. So we started out with something very low-tech in the office by kind of getting a read, some feasibility in humans. We published the first studies back 10 years ago. Then it became that device, which then turned into this current system. It's called Aura, and the company was acquired by Novartis Icahn. And again, I remember when I first had the patent and I started the technology, I was peddling it to all these companies. I said, guys, can you help me solve that problem? You have all these people, all these engineers, and we have something that can do this. But guess what? It's so hard for a big company. They have their own priorities, and I'm not even angry at them now that I've sold them 3 companies, because I understand them. And they would rather pay $500 million for that rather than spend $50,000 early on and develop it, because that's how they work. So again, I was lucky that we were able to find the capital, finance, and ultimately be successful to deliver that technology to that. And I think it's just I appreciate what it takes for a tech transfer office early on to really expose the technology and sell it and pitch it, because that's not easy. And there's a lot of noise that you have to get through in order to articulate the benefits. So it's a huge job, and it's not easy. And we published the final data, which literally in the Journal of Ophthalmology, which showed how much better that technology is and how much happier are the patients. We taught the very first course in ophthalmology at the Academy, which was a huge success. And then ultimately for me, it's great when they send me that, oh, we've done about 500,000 patients now with cataract surgery just from the last couple of years. And I said, wow, that's great. That's why I really put my mind to that, because my effort was how can we impact the most patients at the lowest common denominator. And I'll switch gears now. This is my latest company that we just got FDA approval for something really phenomenal. I thought it was one of the most interesting technologies. It was also pretty hard in terms of what it took, about 10 years again. Everything kind of takes 5 to 10 years, so just take it for granted. If you're 5, you're lucky. 10 years is actually okay. But again, in ophthalmology, as we do a lot of interesting things, we had ignored the advances in interventional medicine and interventional radiology and so forth. People go through your veins and arteries and they put coils in your brain and all that stuff. That's so amazing. But somehow it bypassed ophthalmology. And I started working with some engineers, and we realized that we can do so much more in the eye with those technologies. So again, we felt that it was time for ophthalmology to actually experience that interventional technology, and the perfect place was for glaucoma. And glaucoma is a huge problem, and the problem is that you treat it with medications, just like cardiovascular disease, but when you fail, and when medications fail, which they do most of the time, you go for a bypass. You open, like a cabbage. That was the surgery of glaucoma, and I'll show you. It was a huge surgery where you take out the wall of the eye and you do a huge procedure that I don't want to be done on myself. And so nobody thought of better ways to do that. So the field of glaucoma, before 2000, I was a glaucoma specialist. I'd done those surgeries. They had this draconian, old, established 50-year-old legacy surgeries that were done by the year 2000. That was the standard of care. Once you need surgery, you get a tube implant, which is huge by eye standards, and it requires an hour. And I didn't want to show you the surgery because I wasn't sure was it before or after dinner or cocktail, so I didn't want to have people turn off. But again, entrabeculectomy. We're cutting part of your eyeball, the wall of the eye, and it's a 1-hour procedure and horrible outcomes. 15 to 20% of patients fail every year. When we started innovating, 2010, 10 years later, already quite a few people trying to solve the problem, innovation trickling in. Now 6 options were out there. And today, there's about 9 and probably another 5 on the making. So that field, after 50 years of 2 surgeries, and when somebody told me, Sean, we don't need a better surgery, we already have surgery for glaucoma. That was the 2 cases. Right now, imagine how much innovation is happening in that field. And we got the FDA approval of the first supraciliary stent, which has best-in-class and first-in-class. But when we started out, that wasn't the case. It was very hard to convince people. So what happens here is that really it's almost like a needle stick, and you can see the stent is right in the angle of the eye. This is your iris, this is your cornea, and you can see how the stent is really a tiny little stent, about 6 millimeters, like an eyelash, and we put it into the supraciliary space in a very atraumatic way with that applier, again, interventional technology. And rather than opening up half of your eyeball and doing all kinds of fancy surgery that we glaucoma specialists love to do, we can do that for a few minutes. And no bleeding, no scarring, and very, very elegant procedure that if I had glaucoma, I would like to have. And you can see the surgical procedure here. Here it is, you're going into the eye, and you basically advance the stent through a small incision, less than 1.5 millimeters. And that's the actual editing case. I mean, in this case, it could be a minute, and you're done. No bleeding, no suturing. We've really taken out something that takes a year, an hour to do. We've really reduced it. And the efficacy, once we have that stent, you can see here, it sits in a very privileged part of the eye, right below the sclera. So it's tucked in and doesn't cause any inflammation. And we compared it to just cataract surgery on top of that. And you can see the efficacy was exceptional. And in fact, we got it approved by the FDA without a panel. This was a PMA, one of the long 10-year programs, not a 510K for those that know. And we, best in class, kind of a new category of surgery. It was really surprising. The data was so convincing that control of the disease in terms of intraocular pressure was astounding. The FDA really liked the study design, and they basically approved it without a panel, which really is rare. So as we come, I just wanted to share with you a couple of things I'm working on now, because that's old. We've already gotten it approved, and I like when my colleagues talk about this more than I do, because I like to focus on something else. One of them is printing drugs on the eye, which is piezo micro treatments, where currently we give, any eye drop you give to the eye is basically given with an eyedropper. An eyedropper is something that's been around for 150 years. It's basically a form of a pipette. In the old days, they used to have those pipettes where you take out and you put it in your eye. That's what all the eyedroppers are. They overdose you. In fact, they give you about 8 to 10 times more drug than you need, and that's why many patients have redness, allergy, and the preservative of the eyedroppers are overdosing and causing toxicity on the ocular surface, not to mention some drugs get into your systemic circulation, and through the nasolacrimal duct, they bypass the liver, and it's almost like an intravenous dose. So I can take and put some Timolol in your eyes, and I'll drop your blood pressure. That's what happens now, and that's very well described. Again, I think this new device is exciting because what we use is the printer technology from your inkjet printer. It has piezo vibration that is able to shoot anything very precisely in a collimated way. And the challenge with shooting something in your eye, or I should not say shooting, if the marketing guys were here, they'll say, don't say that. But the challenge of putting something in your eye is that you have to do it before you can blink. And your blink reflex is about 100 milliseconds. So most people immediately blink. So this thing can really go in at about, beats the blink at about 50 milliseconds. So before people realize it's there, and it does it in such a minor way that really you don't feel anything except some wetness. And then it's a device that's also connecting. It's a smart technology device, and it's connecting to your mobile phone. And we know what patient compliance is. We know if they're taking their drugs and whether they respond or not. So that's really exciting. And we're building a whole portfolio of therapeutics now called microtherapeutics, because we can put any of those drugs and really make them proprietary again and make them new and give them a new life, because their safety profile is going to be different. The other technology is, it's in the vein of the previous, which is the interventional technology for glaucoma. We are actually finding that we can use micro-interventional technology now to take care of cataract surgery. And instead of buying a big machine that does the phacoemulsification, which costs about $100,000, we can do 3 pen-like devices to take out the cataract without exposing the eye to any heat and vibration. So we're doing that. Actually, one of the devices got cleared by the FDA a couple of months ago, and we're preparing the commercialization of that technology, which is very interesting. And at the end, I just want to say that it's not only about innovation for private enterprise. Again, for me, it's been lucky because I spent part of my life in academia, and I still spend time with the residents and so forth. But I spend most of my time developing those technologies outside because, unfortunately, you cannot develop them in the universities. I realized that early on, that it's a great place for discovery. You need really processes and industry to develop them. But you can develop outside the private enterprise. And I started several years ago a foundation called Keep Your Sight Foundation, where I donated technology we developed back in the day, which allows people to do a visual field test, which is the same test for glaucoma, where there's 4 million people with glaucoma. Half of them are undiagnosed in the US. And you can take that test that optometrists use or ophthalmologists use in the office, which is the blinking light test and the clicking test. If you guys have seen it, we've actually virtualized that, and you can do it online for 3 minutes, the same test. And we've detected a lot of glaucoma. We screen 10,000 patients a year. I funded the foundation. It's a scalable digital health that we are able to now diagnose people online with a 3-minute test. And we've diagnosed people from Europe that came in and they had brain tumor, because some brain tumors can impact your visual pathways. We've diagnosed glaucoma. It's been phenomenal. So again, I think it's great because I was just tired of us thinking about, oh, well, let's solve the blindness problem, but half of glaucoma is undiagnosed. Well, what are we doing about it? We had a bunch of vans walking around and people doing screenings at 10 patients a day. That wasn't a cost-effective way to do this. So I felt like we can virtualize that technology, and then literally anybody, it's a cloud-based screening technology. Everybody can do it at 3 minutes. And then the other one is really, I'm on the board of the American Society for Cataract and Refractive Surgery. At the foundation there, they have been doing humanitarian cataract missions in the world. But for 20 years or 50 years, we've been doing that, and it's great, it works. It just so happens that 20% of those patients that we operate for cataract have a very advanced glaucoma. And it never occurred to anybody that how we're solving that. We're going to solve the cataract, but they go blind from glaucoma. That wasn't a very good spend of resources even. So we actually created last year the first humanitarian glaucoma surgical mission that used that new interventional technology I showed you, because it's only with this technology that we can do third world surgery for glaucoma. And it was a success. It was a huge success. And in fact, it's going to be probably a paradigm for new humanitarian work. So again, innovation does not have to be a widget. It does not have to be a drug. It does not have to. It could be a foundation. It could be nonprofit. It could be a process. I mean, process innovation. So there's a lot of things one can do. And again, I want to thank you for listening to me, and hope these perspectives are helpful. If you guys want to have any questions, I'll be happy to discuss. It's hard for me to see because the light is in my face. So.
That was an excellent presentation. I'm curious, as an innovator, most of us seem to have or about you as an innovator, but most of us who are wannabe innovators are trying to fake it till we make it. A lot of us have a lot of ideas, and I'm curious how you process your ideas and rank them, and then really make the decision to pursue something. Like if you have a lot of different things that you're interested in, how do you decide that this is what you want to double down on and really pursue?
Right. I think, this is not an easy question because I don't know how other people do it. I think for me, especially when I start doing a lot of parallel projects now, where I'm involved in a few things at the same time, it always starts with the idea, and then I want to pressure test it, and I pressure test it. Sometimes it takes a year, sometimes it takes 5 years, whatever just is an inflection point in your thinking that this has legs. Earlier on, I was very excited about the ideas, but then ideas are not really everything, and I keep saying, a lot of my colleagues come and say, Sean, I have this great idea. I said, okay, well, is it a solution? Because the idea may be great, but it may not be a solution to anything. And then if it's a solution, then you have to think about, well, is that solution actually a product? Because a lot of solutions may not ever be a product, and that takes another set of thinking about it. And then even if you have a product, is that thing a business? Because it may not be scalable. You can just make one of something and not scale it to be a business that can support an enterprise. I think the tech office knows that very well. So I think that I've gotten better through the years, and because of my involvement on the venture side, because of my having been a developer of these technologies and knowing the regulatory and product development pathway, I've come up to the point where I can integrate a lot of that, but each one of them is very difficult. And that's why I think that for me, some things may be intuitive, but really understanding the regulatory complexity is huge. Understanding how you can productize this is huge. So I think, really pressure testing as much as possible without obviously jeopardizing your product or your invention or something new, because everybody is so, especially in the early stages, everybody wants to, feel like, okay, this is a great idea, can I protect it? The patent office, and I think the way the patents work in the US are good, because you can even for very little money spend the money and do your provisional and kind of get a little bit of a, I think, cushion and protection. But then afterwards, really try to, I think, find the people that can help you understand the pros and cons. Because there have been now a couple of things that I didn't do for reasons that, when I looked, were probably the right reasons. And I'm glad I didn't spend a ton of effort on that.
Hi. You mentioned that interventional radiology finally found its way into ophthalmology. And I'm wondering, two questions actually. One, has functional imaging and molecular imaging found its way as well? How far have we gone with that in terms of ophthalmology? Things like fluorescent imaging, intraoperative microscopy, things like that. And also, there's another field that's coming up, which is microbial studies in human beings. We know that the cornea has its own set of microbiome. We're wondering if are there any emerging studies linking the microbiome of the eye with any diseases?
Right. Well, I think that, it's so funny because in ophthalmology, we're so privileged in some way, which could be to our detriment. When you look at psychiatry or issues of depression, I think we're talking next door, it's so hard to really have a firm outcome and know what you're impacting. In ophthalmology, we've been spoiled because we can see it. I mean, the eye and the ability to be able to look at something through microscopy or at the slit lamp. But at the same time, it was a detriment because we were so good at seeing it, we never thought that we needed anything better. So, in fact, through our Genentech studies, when we were doing macular degeneration, the OCT technology, optical coherence tomography, came into being. And in fact, we didn't include it in the study because they didn't have the technology ready for clinical trials. But we did do a couple of studies right after. So with an OCT, we can actually image the layers of the retina now and also the cornea with very high precision. And now it's almost, for everybody, it's a no-brainer why you would use that. But again, when it first started, people said, well, I can see everything. Why do I need an OCT? And it was the way to quantify it too, because we can quantify the edema in the eye that we couldn't do it before. So again, I think we were a little bit disadvantaged because we had so much, I think, an advantage over the other fields. We can see things. At the same time, we didn't feel motivated to explore that much. But right now, that's all changing. And in fact, with us, we did the first thing in the OR, but now there's a whole platform built by companies doing biometry intraoperatively, from intraoperative OCT to many other imaging technologies. So that's going to be the next 10 years. It's going to be really, the microscope is just the platform. The microscope is going to get loaded with, we're the first ones, but it's going to a lot of biometry will go into that. So to me, that's already a reality that will take a couple of years to just become commercially viable. The second reason, I mean, obviously the conjunctiva is full of bug. I mean, there's a lot happening there, and it's a coexistence. Like, for example, dry eye, meibomian gland disease. These are things that we don't even know what causes them, but in fact, it may turn out that microbiome is very important there too. So people are already starting to look into that. I haven't seen a breakthrough to say, hey, this is what it is, but I think it's a very, just like it's in your gut, it's in your conjunctiva. It's very much rich of all kinds of bacteria, and microbiome is very much a part of that ecosystem there. So we just have to see, and it's going to be interesting because I think we're going to learn a ton that we didn't know. And I think dry eye, meibomian gland disease, big things may actually be ripe for some sort of breakthrough with that.
One last question.
What is the current state, and what do you think the future might be for electronic bionic implants, for the retina, similar to what cochlear implants do for deafness?
Right. That is something that we just had one approved recently, Second Sight. My friend Mark Humayan worked on that for many years, and at Duhini. In fact, I was on one of the surgeries when they did. It's really moving forward. I mean, to have an FDA-approved product for a retinal implant, that was a huge achievement. It happened about, I think, 2 or 3 years ago. So Second Sight is a company that's moving that forward. And this is the same thing as computer technology 40 years ago. I mean, we're having a race, the microelectrodes that are maybe 16 now or 64, the maximum. This would be miniaturized exponentially, and it's going to be really interesting. I think that we're just at the beginning of that, and I can only speculate what's going to happen in 10 years. But it's always nice to have a proof of concept, and it's always great when the FDA really approved a product that has very narrow benefit, but it really sets a precedent because it's a true unmet need.